In this lecture, we're going to talk about genetics, and specifically, we are going to be talking about prokaryotic genetics. So I'm going to assume a lot of things about your understanding of general DNA replication and protein synthesis and things like that. If you need to brush up on those things, they are in the textbooks. Not that you're going to have some special recollection of them on exams, but if I'm talking about some aspect of protein synthesis and you don't understand that because you don't remember what transcription or translation is, then my suggestion to you would be to go back into your textbook and review that information, because what I would like to do here is build on that knowledge and talk particularly about prokaryote. So what I'd like to do first is talk about the contributions of microbiology to the field of genetics, and then we'll talk about the specifics of genetics in more detail. First up, a rather unique alga named Acetabularia. And the reason that this microorganism is unique is because this microorganism is not micro at all. They actually are quite large, as you can see from the picture. They're visible to the naked eye, but each of those plant-like structures is actually a single cell. And it's a really interesting cell that's grown into this morphologically distinct plant-like structure, which has a foot that kind of acts like a root structure. It has a stalk, and then it has a cap, which looks somewhat like a leaf. But this, in fact, is a single-celled organism. And a researcher by the name of Joachim Hammerling did some experiments where he was trying to figure out where the information for how to make this cell was, and was it in the cap, or was it in the stalk, or was it in the foot? And he had a hunch that it was in the foot because the foot contained the nucleus of the cell. And so he did a couple of different experiments where he cut the cap off of the tube, and a new cap regenerated, presumably because the information for how to make that cap was found in the nucleus in the foot of the cell. And in order to make that a more plausible explanation, he did another experiment where he removed the foot, and no new foot regenerated, which indicated to him that the information for how to make the foot was not found in either the stalk or the cap. And then he took this one step further and did a really interesting experiment where he took two different species of Acetabularia and he grafted portions of one onto the other to see what would grow. So in the first one, he took this Acetabularia mediterranea and he cut off the stalk and the cap, and then onto the foot he placed the stalk from a second species, Acetabularia crenulata. And what I probably would have assumed is that the cap would grow to look like the cap of an Acetabularia crenulata, but what happened was that the cap of an Acetabularia mediterranea showed up. And so again, this was information that led him to believe that the information for how to make this cell was found in the foot. He did a similar opposite experiment with Acetabularia crenulata, where he cut off the cap and the stalk of those plants and then grafted to them the stalk from the Acetabularia mediterranea, and as he probably expected at this point, the cap of the crenulata grew on top of that. So this was a really interesting experiment done on microorganisms that showed in the 1930s and 40s that the information to how to make cells, the information that we today call hereditary information, was found in the nucleus of these cells. And he was able to do that because he was working with a very large single-celled organism. Prior to the work of Hammerling, a biologist by the name of Frederick Griffith was also doing experiments trying to figure out what was the nature of the hereditary material of the cell. And he had a hunch that that hereditary material was nucleic acids. At the time, it was either thought that the hereditary material of the cell was nucleic acid or protein, and the reason for that was that they had isolated chromosomes, and those chromosomes were made of both proteins and DNA. And so the thought was one of those two things, and in fact, the prevailing thought was that the protein component was the hereditary material of the cell because of the fact that proteins are so much more diverse, and there are, dozens, well, 20 amino acids that make up proteins, whereas there are 4 nitrogenous bases that make up nucleic acid. So he set out to try to answer that question by doing an experiment on some mice and infecting them with a species of bacteria called Streptococcus pneumoniae, which comes in 2 different strains. Remember from our previous lecture, strains are the same species, but they are phenotypically or morphologically distinct from one another. These 2 strains were differentiated by the fact that one strain was capable of making a capsule and the other strain was not. So when those cells were grown on media, the non-encapsulated cells appeared rough, and so they were called the rough strain. And those that were capable of forming a capsule created these larger smooth colonies on the surface of the agar, so those were called the smooth strain. So the difference between the rough and the smooth had to do with the fact that the smooth could make a capsule and the rough could not. Now, what that meant for the mouse host was that if he was injected with the smooth encapsulated bacteria, that capsule would inhibit the mouse's immune system and the mouse would typically die of that infection. So there was a lethal strain of Streptococcus pneumoniae that was encapsulated, and there was a non-lethal or non-virulent strain, which, because it could not form a capsule, was incapable of causing infection in the mice. So Frederick Griffith set up a control where he injected mice with this rough strain, which was non-virulent, and not surprising to him or probably anyone else, the mouse lived. Then he set up a second experiment with what would have been a lethal bacterium, except for the fact that he had killed those bacteria by boiling them. And this is going to become really important because right there, he's done something to indicate to us the removal of the proteins from the experiment. As you know from previous lectures, heat kills by denaturing proteins. So what he has done is killed these cells by denaturing their proteins and not affecting their DNA. And when he takes these dead cells and he injects them into the mice, not surprising to anyone, the mouse lives. So we have this setup where 2 non-virulent types of cells have been shown to not cause disease in the mice. And then Frederick Griffith does something a little bit unusual. He takes the live rough and the heat-killed smooth strain, he mixes them together and he injects them into the mouse, and the mouse dies. So the mouse has died of what should be a non-lethal infection. Furthermore, when Griffith removes the bacteria from those dead mice, he finds live, smooth cells. And then he goes further and takes those live, smooth cells that he's recovered from that second experiment, injects them into mice, and those mice also die. So what he decides from doing this research is that there is some kind of transforming principle inside of these smooth cells that, though they are dead, that they've been heat killed, this transforming principle is able to move from the dead smooth cells into the live rough cells and transform them into smooth cells. And he hypothesizes in 1927 that this transforming principle is DNA. And we're going to see another group of researchers pick up that idea and identify that transforming principle as DNA in a later experiment. First, though, we're going to pit stop on a totally different experiment that happened between the work of Frederick Griffith and the people who picked up on his work in the 1940s, and that is the work of two researchers, Beadle and Tatum, who were awarded the Nobel Prize for their work in 1941, which identified and connected genes and enzymes to one another. And this was, again, going on the assumption that DNA was the hereditary material, and as the hereditary material, it was coding for the production of proteins. It was basically protein recipes. And they did their work on a red bread mold called Neurospora crassa. So again, we see work done with microorganisms. First, that gigantic alga, then down to our Streptococcus pneumoniae, which is a bacterium, and now to a fungus, Neurospora crassa, whose spores were mutagenized by X-rays. Remember from previous lectures that X-rays are non-ionizing radiation that creates free radicals, reactive oxygen species that go in and damage the DNA. So they mutagenized a bunch of spores, and then they mated those spores, because in fungi, spores are the reproductive structures. They mated those with wild type, which is a word that means it grows normally, naturally, exhibiting the normal phenotype. And they crossed those, and then they grew them both in complete media, which would provide them all of the amino acids that they needed, and then in a minimal medium that didn't have amino acids. And what they were trying to do was find mutants that could grow in complete media, but who could not grow on minimal media, because their assumption was for those mutants who could not grow on minimal media, you would have an organism who had a mutation in the gene that coded for that amino acid. And what that would mean is that they would be unable to grow without being fed that amino acid. So they found these mutants, for instance, the middle tube here, and then they determined which amino acid this organism was incapable of producing for itself. So in this case, the spores that couldn't grow on a minimal medium were tested on minimal media with one single amino acid added. So alanine, threonine, tyrosine, methionine, proline, lysine, tryptophan, valine, and isoleucine, when these were added, the organism still wouldn't grow. In the arginine tube, however, the organism did grow, which indicated that they must have a mutation in that pathway and were incapable of producing that particular amino acid. And in the case of arginine mutations, they were able to discover that there were multiple steps in the synthesis of the amino acid arginine, each one governed by a single enzyme, and that led them to that one gene, one enzyme hypothesis, wherein they suggest that every single gene codes for a single enzyme. All right, back to the transforming principle. I know it seems a little weird to go back and forth, but I wanted us to think about these things in chronological order, because we always want to remember that all of these experiments are not happening in bubbles. Things are happening in a particular sequence, and people are adding knowledge to the general dogma of genetics. And so it could be that the work of Beadle and Tatum actually helped with the work that these researchers here, Oswald Avery, Colin McLeod, and Macklin McCarty, did as they followed up and tried to identify that transforming principle. So what they did is they revisited this idea of Streptococcus pneumoniae with this capsule who was heat killed, and tried to figure out what was it inside of that cell that was the transforming principle. And it had to be one of three things. It had to either be protein or DNA or RNA. So they took Streptococcus pneumoniae, they heat killed them in the same manner that Frederick Griffith did, and then they removed the capsule components from solution. So they take this solution, and as a control, they add the DNA, RNA, and proteins to one tube. They use a protease in the second tube, and the protease, as its name suggests, digests protein. So all of the proteins would be digested, but the RNA and the DNA would still be present. So after we have the control satisfied in just a few moments, the second tube, the protease degraded tube, will give us an idea of whether it's proteins that are the transforming agent, because if it is, then there shouldn't be transformation in that tube. In the third tube, ribonucleases are added in order to degrade the RNA, right? RNA is a ribonucleic acid, so a ribonuclease degrades RNA, and in this sample, proteins and DNA are still present. So if it's RNA that is the transforming principle, then this should not allow transformation. In the third experimental tube, deoxyribonucleases are added. Those deoxyribonucleases, of course, degrade deoxyribonucleic acid. So in this tube, proteins and RNA are still present. So if it was the DNA that is the transforming principle, then this tube should have no growth, right? So in the first tube, transformation should occur. That's the control. That's just telling us that we haven't, through the process of heat killing and removing capsule components, done anything to negate transformation. In the first tube, we want to look and see if proteins are the transforming principle. If they are, no transformation will occur. In the second experimental tube, we want to see if RNA is the transforming principle. If they are, no transformation will happen in that tube. In the third tube, we want to see if DNA is the transforming principle. And if it is, then there will be no transformation in that tube. And lo and behold, our control is satisfied. We have transformation in the first tube, which tells us that transformation occurs in the absence of proteins, so it's not the proteins doing it. In the third tube, we have transformation in the absence of RNA, so that tells us RNA is not the transforming principle. And then finally, in our fourth tube, there's no smooth cells. Transformation of the rough cells that were added did not occur, and so transformation does not occur without DNA, which indicates to us that DNA is the material directing the synthesis of the capsule. So now we see DNA, right, the gene directing the synthesis of the enzymes for arginine production in molds, and we see DNA as the material that is directing the production of the capsule. And finally, in 1952, Alfred Hershey and Martha Chase unequivocally demonstrated that DNA was the hereditary material of the cell. And they did that by doing a very, very simple experiment where they took bacteriophage, which are viruses that infect bacteria, and they radioactively labeled the two components of those bacteriophage, right? They're so simple, they're not enveloped viruses, though they have a complex morphology. That complex morphology is all protein, and all of that protein surrounds DNA. So it's got to be either the protein or the DNA that is infecting bacteria and causing those bacteria to make baby viruses. So in one experiment, Hershey and Chase radioactively labeled the DNA with phosphorus and allowed those radioactive bacteriophage to infect bacteria, and then they looked for where the radioactivity had gone. If the radioactivity had gone into the cells, they would know that it was the DNA that was directing the production of new viruses. If the DNA stayed in solution with the bacteriophage, then they would know that it wasn't the DNA that was directing the synthesis of these bacteriophage. And when they did this experiment, notice the bacteria infected with the phage containing that radioactive DNA were inside of the cells. So this gave them evidence that DNA was the hereditary material, but they backed that evidence up by doing a second experiment to disprove the protein by using radioactively labeled proteins. And in this way, they used sulfur, which is never a component of DNA, so that they could isolate those two molecules from one another. So they radioactively labeled the sulfur component of the protein, allowed the bacteriophage to infect the cells, and then looked for the radioactivity and saw it in the solution in the supernatant, not in the cells. And so these two experiments, which were done in 1952, as I mentioned, demonstrated finally, and sort of were the mic drop moment of the age-old question, what is the hereditary material of the cell, that had been asked over and over again since the days of Gregor Mendel and his pea plants. What is the hereditary material? The hereditary material is DNA, and that DNA was shown to be the hereditary material through the work of many microbiologists. So we want to give those microbiologists their due. So DNA is the hereditary material of all cells, and that goes for humans, plants, fungi, protozoans, algae, E. coli, everybody, right? Everybody uses DNA as the hereditary material of the cells. Now, the one exception to that is viruses. You might remember from our earlier lectures that viruses can have an RNA genome. They can even have an RNA genome that they convert to DNA and then back to RNA. But cells all use double-stranded DNA as their hereditary material. And there's a lot of variability in terms of how much DNA different organisms have. Human beings have about 3 billion base pairs in their hereditary material. E. coli have about 4.5 million base pairs in their DNA. Plants, surprisingly, maybe have lots and lots of DNA. A lot of them are tetraploids, which means they have multiple copies rather than just being diploid. But the take home here is that DNA is the hereditary material of all cells. That's thanks in great part to the work of people working with microorganisms. One last nod to give to the microbiologists before we move into talking about genetics is the idea of how DNA is replicated. And there were, in the 1950s, 3 prevailing theories for how DNA was replicated in cells. One was called the conservative theory, the second was the semi-conservative theory, and the third was the dispersive theory. In the conservative model, it was proposed that parent DNA remained associated in one DNA molecule and a new daughter strand was made and given to daughter cells. So conservative in that that parent double-stranded DNA molecule would remain together throughout multiple generation times. The second theory, the theory of semi-conservative replication, said that the parent DNA served as a template for the daughter strands by opening up and allowing for a second strand to be made adjacent to each of those parental strands of DNA, so that after one round of replication, you would end up with each double-stranded molecule having one parent strand of DNA and one new strand of DNA. So in the conservative replication, one single-stranded molecule has all of the parental DNA and one single-stranded molecule has all of the new DNA. In the semi-conservative theory, each of those daughter strands ends up with one old and one new strand of DNA. In the third theory, the dispersive model, all of the resulting DNA strands have regions of double-stranded parental DNA and regions of double-stranded daughter DNA. So there's no real pattern to the way that these are replicated. So in order to determine which of these replication theories could actually be supported scientifically, 2 researchers by the names of Matthew Meselson and Franklin Stahl devised an experiment where they would look at the DNA and watch it move as the cell went through rounds of replication. And the way that they were able to differentiate the DNA strands that were new versus the DNA strands that were the older parent DNA was by radioactively labeling the nitrogenous bases of the parent DNA. And that made them a little heavier because nitrogen 15 is a little bit heavier than atmospheric nitrogen, which is 14. So they could differentiate based on this experiment the weight of the DNA. So the heaviest DNA, the parent DNA, would fall much lower in these tubes. And if conservative replication was correct, then you would always only have this very heavy DNA and very light DNA. There'd be no mixing of the DNA, right? That first theory says that after one round of replication, you end up with that conserved parental double-stranded DNA and a new double-stranded DNA. So the blue line would be the one that was the parental DNA, the red line would be the one that was the the new DNA. If semi-conservative replication was correct, then the parental DNA would open up and combine with a new DNA, so you wouldn't end up with heavy DNA or light DNA. You'd end up with a hybrid DNA molecule that existed in the middle. And then once you went through a second round of replication, you would still have those hybrid pieces in the middle, but you would also have the light pieces who were made from the light half of that hybrid molecule of DNA. If the dispersive model was correct, then you would begin with heavy DNA, move to hybrid DNA, and always have hybrid DNA after that. So they set up these predictions and allowed cells to undergo rounds of replication, having had heavy DNA and adding to it light DNA, and what the results showed was semi-conservative replication. That these cells of E. coli were in fact using the parental strand of DNA, opening it up, using it as a template, and adding to each of those parental strands one new strand, creating hybrid molecules of DNA. And then after a second round of replication, those hybrid molecules were opening up. Each parental half of the hybrid molecule would remain hybrid, but each new half of those hybrid molecules would become light as its second strand was replaced with new light nitrogen. So again, lots of experimentation to think about, but I don't want us to ever forget that microbiology doesn't exist in a vacuum, that the things that we know today, we know because of the hard work and the combined efforts of many different researchers over a long period of time. And I think that's important both to acknowledge those contributions and also how important it is that scientific information is meticulously organized and shared with other people so that the knowledge can become cumulative. So fast forward to 2020, and what we know is that DNA serves as the recipe for proteins as it is transcribed into messenger RNA, and those molecules of messenger RNA are translated into proteins. So what we're going to focus on for the rest of this lecture is how prokaryotes particularly transcribe and translate their genes in order to produce proteins. In order to do that, we're going to talk about three basic topics. We're going to talk about the expression of genes, which is just another way of saying protein synthesis. How is the DNA transcribed and translated in proteins uniquely in bacteria? We're also going to talk about a phenomenon called genetic recombination, where microbial cells can share information with one another. And then we'll talk just a little bit about DNA replication, because DNA replication really is so similar between eukaryotes and prokaryotes. We're not going to spend a lot of time talking about that topic. We're going to primarily focus on gene expression and recombination. As we talk about microbes sharing genetic information with other organisms, I want to distinguish between the sharing of genetic information with your offspring and the sharing of genetic information with organisms in your population. And DNA replication is the way that we share information with our offspring, and we call that vertical gene transfer. It's the transfer of genetic information vertically through a cell line from parent to offspring. Horizontal gene transfer, on the other hand, is the sharing of genetic information not with your offspring, but with other members of the population. So we're going to talk primarily today about horizontal gene transfer because you've talked a bunch about vertical gene transfer in general biology. Then we'll wrap up by talking about gene expression as it exists specifically in microorganisms. So the funny thing about microorganisms is that we still can have the idea of genotype and phenotype even though all microorganisms are by definition haploid cells. They're haploid, meaning that they have one single chromosome with one gene for every trait that they have. So they have a genotype, which expresses itself as a phenotype, right? The genotype is the gene that you have, the phenotype is what that gene makes you look like. And you would imagine, especially when you think about dominance and Gregor Mendel and his pea plants and dominant traits and recessive traits and all of those things, that microorganisms, because they have only one allele for every gene, would have one possible genotype. But phenotype is not just reliant on what genes you have, it's reliant on whether or not those genes are being expressed. And it turns out that microorganisms do express different genes in different environments. You might remember we talked earlier on in the semester about motility and how certain organisms can turn on the production of multiple flagella in order to respond to an environmental change. We've also mentioned biofilms. Biofilms are typically genes that are turned on in response to environmental changes. We've talked about sporulation as a response to environmental changes. So the genes that a microbial cell will turn on varies with the different environmental conditions. For instance, here we have Serratia marcescens. Serratia marcescens at room temperature, 25 degrees Celsius, will turn on the genes to produce a red pigment. At 37 degrees Celsius, it will not activate that gene, and so it will be colorless in those environments where the temperature is too high. So we call that a temperature-dependent pigment. Temperature being a change in the environment, which now changes the phenotype of the cells. So though we don't have multiple forms of alleles in haploid cells, we do still have differences between genotype and phenotype in these bacterial chromosomes that are very much like yours. They are double-stranded complementary DNA molecules where A's pair with T's and C's pair with G's. They undergo the same semi-conservative replication that Messelson and Stahl identified in 1958. And though they are like yours in many ways, they are unique. As I mentioned earlier, they're always haploid, their chromosomes are circular, and unlike your DNA, which has about 98% of the nucleotides considered to be non-coding, what used to be called junk DNA, bacteria have an average of 12% of their DNA non-coding. So though they have a much smaller genome, it appears that much more of that genome is utilized to direct the synthesis of proteins, right? So our genome is about 3 billion nucleotides, bacteria about 4.6 million. We've got 30,000 genes. They've got about 3,200 genes. So they have smaller genomes. More of their genomes are dedicated to the production of proteins, and E. coli can replicate that 4.6 million base pair genome in about 46 minutes, which just so you know, is about 1,000 base pairs per second. So they're able to replicate very, very quickly. As you know from previous lectures, in addition to the bacterial chromosome, bacteria most often also have small circular self-replicating pieces of DNA called plasmids. And we talked about them very briefly early on. We're going to talk more about them today, and we'll revisit the topic of plasmids later on. Today we're just going to talk about 3 kinds of plasmids. The first is called an F factor, or a conjugative plasmid. The F factor has the genes on it which will code for the production of a sex pilus and the transfer of that plasmid DNA into another cell. And we're going to talk a lot about F factors in this lecture. Dissimilation plasmids are a group of plasmids that contain genes to catabolize a variety of different food items, including octane, methane, ethane, arsenic, TNT, weird things like that. So though the chromosome may not have the genes to catabolize those things as food, these dissimilation plasmids will bring with them the genes that will allow organisms to funnel arsenic into their catabolic pathways, for instance. And then finally, a third type of plasmid is something called an R factor, which codes for resistance to antibiotics. And that's going to be one that we pick up later on in much more detail in our last unit of lecture. We're going to move on to gene expression, but of course, because gene expression involves the transcription of a gene, we're still going to be referring to DNA a little bit. One thing that I want to say about gene expression that is unique to prokaryotes is the fact that because the prokaryotes lack a nucleus, transcription and translation can occur simultaneously. As soon as an RNA transcript comes off of its DNA template, ribosomes can bind to that messenger RNA and start translating the protein, which means that you can have a much faster response to an environmental change. So if you want to change your phenotype in response to an environmental change, you can do that much more quickly if you're a prokaryote. The other difference between gene expression in prokaryotes and eukaryotes is that prokaryotes do not modify messenger RNA transcripts like eukaryotes do. And that may or may not be something that you talked about in general biology. It's not something that we're going to talk about anymore right now, just to say simultaneous transcription and translation, and then also to say that there's no modification of messenger RNA. As soon as it comes off of the gene, it's ready to be translated into a protein. I mentioned earlier that bacteria can regulate gene expression and change their phenotype in response to environmental changes, even though they only contain one allele for each of their genes. And they will do that in a variety of different ways, by controlling whether enzymes are on or off, or by controlling whether or not a particular gene is transcribed or a particular messenger RNA is translated. And depending on how strict the control the microbe wants, it can go anywhere from having an enzyme that's never controlled to an enzyme that's so heavily controlled that even the transcription of the gene is controlled. Some of this will sound a little familiar to you because we've actually mentioned the control of enzymes when we talked about metabolism. We talked about allosteric inhibition and how binding something to the allosteric site of an enzyme can cause a conformational change in the active site of that enzyme and render it on or off based on the activity at the allosteric site. And so what you'll see at the first level of enzyme regulation is that you have a gene, for instance, gene B, that is always transcribed, always translated, but the enzyme activity can be regulated based on a binding of a third molecule to the allosteric site of the enzyme. And so the allosteric binder actually can serve to turn on and turn off this enzyme. And we call that post-translational control because It happens after or post the translation of that enzyme. Now, when there's no control exhibited at all, you have what's called a constitutive enzyme. So these are housekeeping enzymes, enzymes that are involved maybe in glycolysis and things like that. And those are genes that are always transcribed and always translated. And when they're translated, they're on. And they don't get turned on and off, and so they're always doing the job of converting whatever substrate they have into their product. So we can have no control, which you can see here in gene A. We can have post-translational control, which is seen here with gene B. We can have translational control, where gene C is transcribed, but it is not translated. Now, that's not something that we're going to talk about in this lecture, but we are going to talk about strategy D, which is the transcriptional control of this enzyme. So not even the transcription of that gene is allowed unless certain environmental criteria are met. Now, one really cool way that microorganisms regulate their genetic activity is by sensing cell density, and that involves the constitutive production of proteins which are used as name tags, for lack of a better word, where bacteria can throw them out into the environment and give other bacteria an idea that they're around. And at low density of cells and low density of this proteins, bacteria will exhibit these individual behaviors, but when the level of these proteins in the environment gets high enough, the cells will turn on different genes and exhibit group-like behavior, right? Almost exhibit a multicellularity to their behavior. And one big thing that is known about quorum sensing is that it is involved in the sporulation of spore-forming bacteria, because as cells increase in density, there's a scarcity of resources and a scarcity of space and environmental constraints that might tell the cell, you know, it's not time to reproduce right now, it's actually time to sporulate, to go into this protective endospore and ride out this crowded, terrible environment we're in. Another thing that's controlled by quorum sensing is the formation of biofilms. So planktonic cells might be swimming around in the body, and when they sense that there are enough of them to actually make a biofilm, which is a huge endeavor, then they will turn on the genes for producing a biofilm. Another really nasty example of quorum sensing causing a change in behavior is found in Pseudomonas aeruginosa, who, in order to avoid host defenses, will not transcribe or translate any of its virulence genes unless it knows that there are enough Pseudomonas aeruginosa in the environment that they can actually overwhelm the host defenses and cause disease. So they will hide out, be quiet, not do anything to ruffle the feathers of their host, which would cause the immune system to come into the area until there are enough of them there that they can actually mount a productive attack against the host defenses. So quorum sensing is the ability to sense cell density and then modify your behavior based on a high cell density. And what that modification means is exactly what we were talking about in the last slide. It means turning off certain genes and not expressing those genes, and turning on the expression of other genes, all based on the fact that you've got lots of friends in the environment. Now that we understand how bacteria and other prokaryotes can modify their genetic expression and change their phenotype based on transcriptional control, we're going to switch gears to the final topic for this lecture, and that is the phenomenon of horizontal gene transfer. which is probably a completely novel concept to you because it's not typically something that's talked about in general biology. You'll remember from the beginning of this lecture, though, that horizontal gene transfer happens when a bacterial cell shares information with a member of its population, not with its offspring, but with a member of the population that it is living amongst, and that can happen within a species. Pseudomonas aeruginosa can share with Pseudomonas aeruginosa, and it can happen among species. Pseudomonas aeruginosa can share with Escherichia coli or Staphylococcus aureus. So this is not something that is species specific. It is something that is occurring amongst lots of different prokaryotic cells all the time. And it comes in three basic forms. The first, transformation, should sound familiar to you. That was what Frederick Griffith was studying, right? He called that molecule, whatever was getting from the dead smooth Streptococcus pneumoniae into the live rough Streptococcus pneumoniae, was a transforming principle. And so transformation is the uptake of just fragments of DNA from solution that are brought into the cell. Transduction is the movement of DNA into a cell that is mediated by a virus. So here you see a bacteriophage attached to the surface of a cell, and it's injecting that same piece of pink DNA into that cell. And then conjugation involves the F factor that I mentioned earlier in this lecture and the movement of DNA through the pilus, which was actually something we talked about way back in the lecture where we talked about prokaryotic cells. So conjugation involves cell-to-cell contact and the F factor. Transduction involves viruses, and transformation involves the movement of free DNA in solution from one cell to another. The cell that is providing the DNA is called the donor cell. The cell that is receiving the DNA is called the recipient cell. And this process of horizontal gene transfer, which sends DNA from a donor cell to a recipient cell, results in what are called recombinant cells, cells who have now combined DNA from two different sources themselves and the donor cell. And we're going to talk about each of those three procedures in more detail, but what I wanted to do first was just talk about how to make that DNA once it gets into the cell useful, right? Because if you're just talking about a piece of DNA floating around in solution, how does that DNA do anything once it gets into the recipient cell and actually become part of the machinery of the cell that can then be passed on to its progeny, its daughter cells? Because if this is a beneficial piece of DNA, let's say this is a gene for antibiotic resistance, that's something that that bacterium is going to need to express in order to actually have the phenotype, and it's something that the bacterium is going to want to pass on to its offspring if it's ever going to survive in this environment where antibiotic resistance is necessary. So I wanted to take a look just very quickly at two mechanisms for making that DNA functional in the recipient cell and actually making that recipient cell not just recipient but recombinant, because a recombinant cell has that DNA somewhere useful, right? Either it's in its chromosome or it's existing as a plasmid. So the first of those was transformation, which involves the uptake of naked or free DNA in solution. If that fragment of DNA is some portion of the chromosome, of course, it can be taken up and brought into the cell, right? Which is something that cells will do. Cells have DNA binding proteins on their surface, and they will take those fragments of DNA up, and if nothing else, they will use them in order to make pieces of DNA and save themselves the work of having to create those nucleotides. But sometimes a recipient cell will take up these DNA fragments, and they're shown here with that typical genetics paradigm of big A's and little a's to show you that these genes that have been taken up with this new fragment of DNA are just alternate forms of genes that are already encoded in the chromosome of the recipient cell. So much like we have brown eyes and blue eyes, there might be two different genes that would code for a beta-lactamase gene that would make a cell antibiotic resistant. So maybe the big A is susceptible to certain kinds of antibiotics, but the little a is not susceptible. So this fragment of DNA comes in, it lines up with the complementary portion of the chromosome, and then Rec-A proteins allow that recombination to occur, and then you get this recombined recipient cell. The situation is much, much simpler if you're talking about the movement of a plasmid into a recipient cell, because plasmids are completely functional pieces of DNA that can exist separate from the chromosomes. They have their own origins of replication. They do not need any of the machinery of the chromosome in order to be passed on to the offspring, and they don't need any of the chromosome in order to be expressed. And a lot of times plasmids come with novel genes, genes that are not analogous to something that's in the chromosome. So that's shown here as an E. So again, the proteins on the surface of the cell would bind this DNA, take it into the cell, and as soon as that plasmid gets into the cell, it can be expressed, the genes on it can be expressed, and it can be replicated as the cell undergoes binary fission. So transformation is the movement of naked or free DNA that's in solution to a recipient cell who then becomes recombinant. Transduction is a method of moving DNA that involves a virus. So what basically happens is a virus infects a cell, it makes a mistake, and accidentally picks up a piece of that cell's DNA, and then when it packages all of the DNA into new bacteriophages, it accidentally puts some of the first bacterium's DNA into it as well. So if you remember back to the stages of viral replication, we have attachment, biosynthesis, maturation, and release, right? Well, attachment and penetration very quickly are followed by a dismantling of the host chromosome. So the host chromosome is chopped up in order to stop the host cell from doing anything bacteria want, and instead becoming little virus-making factories. And so then biosynthesis of the portions of the viral genome and parts are made, and then they're packaged together into bacteriophages, and part of that packaging is to shove that viral DNA into the head of the bacteriophage. But sometimes, because it's crowded and there's a lot going on, instead of packing bacteriophage DNA into the head of the bacteriophage, it packs a piece of bacterial DNA in there. So that's shown here as a little blue piece of DNA for the viral DNA, and a little pink piece from the donor bacterial cell. And when in step 4, this new phage attacks a new cell, the recipient cell, it injects into that cell both the viral DNA, which is shown in dark blue, and a little piece of bacterial DNA, which is shown in light blue in this new pink chromosome. So transduction is the delivery of DNA, which is mediated by a virus. Finally, we have our third method of recombination, which is conjugation, which is always mediated by that F factor. So when cells have an F factor, we call them F plus cells. And what F plus cells have the ability to do is make a pilus and send the F factor into a recipient cell. So you can see that happening here. The F plus cell encounters the F minus cell, produces the pilus, and then in step 2 sends the F factor through to that cell, and then disengages from the cell. And at the end of this procedure, you end up with 2 F plus cells, indicating to you that the F factor has been copied and shared with that recipient cell. So the result of the mating, as it's called, between an F plus and an F minus cell is for both cells to become F plus. So now you have 2 cells that are capable of making a pilus and sending a piece of DNA into an unsuspecting recipient cell, which really, to be honest, is not beneficial. It's energetically expensive, and it means that you're expending energy on something that's completely useless to you. So this is not ideal. However, there are some variations on this F plus and F minus scenario that do end up creating beneficial situations for the recipient cells. One of those is shown up at the top here, where we have an F plus cell who has integrated the plasmid into their chromosome and become what's called an HFR cell, a high frequency of recombination cell. The reason this is no longer called an F plus cell is because it's going to do something a little bit different when it encounters an F minus cell. And that happens because as this HFR cell encounters an F minus cell and begins to create the pilus and start sending through the F factor, it starts to send through the F factor, and then it starts sending a piece of its chromosome over with it. And so you end up with a recipient cell who is not F plus, but he's not really F minus either, because he does have a bit of this F factor, so he's called an F prime cell. And what he gets out of the situation, possibly, is that in addition to having a portion of the F factor, he might get a useful portion of the donor cell's chromosome. So in the case of an HFR and an F minus cell, you end up with an HFR and an F prime, which just indicates he's a little different. He's not F minus, he's not F plus, he's not going to make a sex pilus, and he may not have any functional information because he may not have been connected to the donor cell for long enough to actually get an entire copy of a gene, because it takes time for those genes to be copied and transferred through the pilus and into the recipient cell. But the thing to realize, if you look at the bottom here, is that when the HFR cell starts to transfer, the origin of the transfer of the F factor is right in the middle of the plasmid, which means he starts to transfer half of it, and he doesn't have time to transfer the entire chromosome in order to transfer all of the F factor. In fact, it would take about an hour and a half for this process of moving the half of the F factor, the entire 4.5 million nucleotides of the E. coli, and then the other half of the F factor for this recipient cell to become F plus. So HFR and F minus does not yield F plus cells because they're not connected to one another for long enough for that to occur. Now, the kind of interesting thing that they've been able to do because of the transfer of DNA from an HFR cell to an F prime cell is they've actually mapped the chromosome over that 100-minute timeline that it takes for the initiation of the movement of the bacterial chromosome until its completion. So you can see here, it starts at the origin of transfer and then moves into these genes, and you just follow the clock. So Pro-A and B are first, then PAB-B, and then gyrase, and all of these different genes. And then one of the very last genes to be transferred over is methionine. So kind of cool, but functionally for this F prime cell, nothing's really changed. It's got a little bit of chromosome, which may or may not be useful to it, and it's got a bit of the F factor. The only real change is when an F plus cell encounters an F minus cell. There also will be a summary table in the lecture notes of how different F plus and F minus and F prime and HFR cells interact with one another so that you can understand those processes well.